Next speaker is Eva Rodriguez Traver. She's going from the Cajal Institute, and she's going to talk about effect of mutations in the GBA1 gene in iPSC-derived dopaminergic neurons derived from Parkinson disease patients.
First of all, thanks to the organizers for allowing me to present our results about the effects of mutations in the GBA1 gene on iPSC-derived dopaminergic neurons from Parkinson's disease patients. There are several mutations that have been related with Parkinson's disease. Indeed, mutations in GBA1, which encodes for the glucocerebrosidase 1, are considered the strongest genetic risk factor for Parkinson's disease. Moreover, mutations in this gene are related with an early onset of the disease and an increase in cognitive impairment and dementia. There are different effects related with GBA1 mutations and Parkinson's disease. If this enzyme is not located in the lysosome where it has to function, it's not going to be degraded, the glucocerebrosid, and then this compound will accumulate. And this accumulation stabilizes the formation of oligomers of alpha-synuclein. Moreover, it can directly affect to the conformation of alpha-synuclein. And finally, altogether gives rise to the accumulation of fibrils of alpha-synuclein. Moreover, these oligomers of alpha-synuclein also impair the trafficking of the enzyme from the endoplasmic reticulum to Golgi, where it has to mature to acquire its functionality. So then we will have less active enzyme in the lysosome and more accumulation of glucocerebrosid and of fibrils of alpha-synuclein. But there are other effects related with the general homeostasis of the cell and GBA1 mutation. Indeed, in a collaboration study led by Dr. Rosario Moratalla, using fibroblasts with the N370S mutation, we have observed that they have an impairment in autophagy, with accumulation of autophagosome, an increase of the endoplasmic reticulum stress, and accumulation of dysfunctional lysosomes, and an accumulation also of cholesterol and swollen and dysfunctional mitochondria. Altogether increases the cell death. I don't know how to do it with the pointer. Okay, no. Okay, so in order to have a human cellular model to study Parkinson's disease, we have used the induced pluripotent stem cell technology, or iPSC technology. This technology allows us to reprogram somatic cells from control MPD samples, into iPSCs, which we can then differentiate into the cellular type that we want to study. In our case, dopaminergic neurons, to characterize disease phenotypes and to identify molecular targets. Taking everything into account, we wanted to generate and characterize iPSCs derived from fibroblasts of PD patients carrying heterozygous mutations in the GBA1 gene, the most common ones, the N370S and the L44P, as well as from healthy controls. And we also wanted to generate and characterize dopaminergic neurons from iPSCs to study cellular, molecular, and functional phenotypes caused by GBA1 mutations. First of all, we isolate fibroblasts from skin biopsies. We have different samples, 3 controls, and 2 PD samples with the N370S mutation and 3 with the L44P. Once we have the fibroblasts, we reprogram them into iPSCs using the Sendai viral vector system, which is a non-integrative system. We use the classical reprogramming factors, OCT4, KLF4, SOX2, and CEMIC, and we obtain these colonies with this genuine morphology. They are flat, compact, with sharp edges, and a high nucleus cytoplasm ratio. We culture these iPS cells onto mouse embryonic fibroblasts. Once we have our iPS cells, we perform different genetic quality control assays to determine that they have a normal karyotype, to identify its genetic fingerprinting, and to confirm the GB1 mutations. Moreover, as we are using a non-integrative reprogramming system, we could determine the clearance of the reprogramming factors and sendai genome in our iPS cells, as you can see by the absence of positive band in our samples, after 5 to 10 passages, using RT-PCR. Then we wanted to characterize our iPS cells, and we wanted to study their pluripotency, and we used different markers. The nuclear transcription factor nano, the membrane proteins TRA160 and 81, and SSEA3 and 4. We also determined the expression of endogenous core pluripotency markers OCT4, SOX2, and nano, and we could observe that all our samples express these markers. We haven't found differences in this expression among genotypes. To confirm the pluripotency of our iPS cells, we differentiate them into the 3 germ layers using embryoid bodies. Then we characterize the populations that we have using different markers: alpha-fetoprotein, which label fetal liver for endoderm, desmin, which label muscle for mesoderm, and PAX6 and beta-3 tubulin, or TU1, for ectoderm. All our iPS cells were able to differentiate into the 3 germ layer, and we didn't observe qualitative differences in this process for any genotype. After the characterization of the iPS cells, we differentiate them into dopaminergic neurons using a floor plate-based protocol based on the addition of small molecules for the dopaminergic specification and neuronal differentiation and maturation. For some experiments, we maintain the cells during 90 days. During the differentiation process, we analyze the expression by RT-qPCR of different markers, as the neural progenitor marker, the immature neuronal marker, the neural progenitor marker PAX6, the immature neuronal marker beta-3 tubulin, the mesencephalic progenitor markers LMX1A and B, FOXA2 and NEURON, as well as the dopaminergic markers tyrosine hydroxylase, vesicular monoamine transporter 2, dopamine transporter, and the potassium channel GIRK2. We also analyzed the expression of FOXA2 and with the vesicular monoamine transporter 2 by immunocytochemistry after 54 days of differentiation. And when we quantify the numbers of cells that we have in our cultures, we observe that GB1 mutations do not alter dopaminergic neuron generation. And we observe that we have between 12 to 18% of T8, 24 to 39% of TU1, and from these TU1 cells, we have 47 to 56% of T8. Due to the importance of alpha-synuclein in the pathology of Parkinson's disease, we analyzed the release of alpha-synuclein in the cell culture supernatant by ELISA. And we could observe a tendency for a higher release in PD samples compared to the controls. However, we need to normalize this data by cell number. Moreover, we wanted to study the functionality of our neurons, and for this, we determined the dopamine release by ELISA after 24 hours in basal conditions and after stimulation with potassium chloride during 20 minutes. We quantify the numbers of Th positive neurons to relate the concentration of dopamine per Th cell, and we observe that oral samples release dopamine. We need to repeat these measures, but for the moment we didn't find significant differences. And finally, we wanted to study the electrical activity of our neurons, and for this we used patch clamp technique in collaboration with Dr. Rosario Moratalla. We have also injected Alexa 488 for future morphological analysis. When we analyze the action potentials, we observe significant differences after 82 days in PD samples compared to the controls in the percentage of cells able to fire action potentials. However, this effect almost disappeared after 90 days. We also analyzed the characteristics of the action potential, and we just found a significant difference in the N370S samples after 82 days compared to the controls. But this effect disappeared after 90 days. Finally, analyzing the firing rate for a determined injected current, we observed that N370S samples presented higher firing rates compared to the controls. And this may indicate that these neurons are more excitable. To conclude, we have generated iPSL colonies from using sendai viral vectors, which had a typical iPSL morphology, presented a stable karyotype, were free of dopamine factors and sendai genome, expressed pluripotency markers, and were able to differentiate into cells from the 3 germ layers. We are differentiating dopaminergic neurons from these iPSLs, which express markers of mesencephalic dopaminergic neurons, such as LMX1A and B, FOXA2, NEUR1, Th, VIVA2, and GIRCA2. These neurons release dopamine and have electrical activity, and we are using these neurons to study molecular, neurochemical, structural, and electrophysiological phenotypes caused by GBA1 mutations. Our first results suggest that N370S neurons present a hyperexcitable phenotype. Thank you for your attention, and thank you to Carlos Vicario's laboratory members, and to our collaborators, especially to Dr. Sari Moratalla and our sponsors.
Thank you, Eva. Rafa.
Very nice. I have a question regarding the hyperexcitability. So I have two questions. One, if you have any idea what could be underlying the mechanism underlying. And the other thing is in any in vivo animal model, if you have actually a connection with that.
Yeah, well, there's a paper, I think they use mouse, where they inhibit the proteasome and they observe that the dopaminergic neurons are more excitable also. So as glucocerebrosidase 1 can affect the autophagy and maybe have some impairment also in other degradation pathways, maybe the proteasome, maybe this is the effect that, for example, it can be that the channels in the membrane is degrading properly, so then these neurons are more excitable. And yeah, basically this, I think, and because this glucocerebrosidase affects the lipids of the membrane, may affect this. So.
Okay, very nice.